Hello and welcome to the MedWire News podcast. For this edition, we spoke to Professor Mel Greaves from the Institute of Cancer Research in London, UK, about his proposed hypothesis for the etiology of childhood acute lymphoblastic leukemia.
There's been controversy and debate for about 100 years as to what the cause might be of childhood lymphoblastic leukemia, which is the major type of cancer in children. And for most of that period, there's been a suspicion that infection might be involved. And what this current theory or hypothesis suggests is really to invert the whole problem and to suggest that the issue in developed societies is paradoxically a lack of infection. Now, there's some complicated biology here, which I'll try to make relatively simple. We've spent many years, 2 or 3 decades actually, unpicking the biology. And what happens in childhood leukemia is that a mutation arises in blood cells in utero, in the fetus, which generates a covert, clinically silent, pre-malignant clone at quite a high rate, actually, 100 times the actual leukemia rate. Those clones sit there for up to 15 years, but they can be triggered into overt clinical leukemia in response to infection, in fact, in response to common infections. But what we've unpicked with our epidemiology colleagues and with mouse modeling is that will only happen, that infection will only trigger lymphoblastic leukemia in children who have, paradoxically again perhaps, a deficit of infections in the first year of life. And the explanation for that turns out to be that the immune system in newborn children, as in mammals generally, the immune system network requires exposure to benign and beneficial bacteria in order to regulate itself correctly when it's subsequently challenged, when children go to school, for example. So the first year of life turns out to be critical, and the fundamental risk, so to speak, in children who develop lymphoblastic leukemia in the UK and elsewhere is that these are children who have a deficit of these common exposures early in life. And they're not pathological exposures with dangerous infections, but benign and beneficial bacteria.
So exposure to benign infection appears to be crucial for reducing the risk for developing all. But do we know which microbes are the main players here?
We don't know exactly what microbes are important here, but what the epidemiology suggests is if you look for correlations with known pathological infections early in life, there's no correlation at all with risk of leukemia. The correlation is with things like if you go to play groups in the first year of life, you're protected. If you are breastfed, you are protected from this cancer. If you have cesarean section versus vaginal birth, you're more at risk. And what those all look like, those are the routes by which infants acquire their natural gut microbiome. So those are the organisms we're interested in. And the microbiome is quite complex. It can even be small parasites, but it can certainly be viruses and different bacteria. But maybe the diversity of those species makes a difference. So there's increasing understanding that the diversity, particularly the bacterial component of the natural microbiome, these are beneficial, benign bacteria, primes the immune system.
And what about vaccines? Do they have a role in this process at all?
The question about vaccines is a very interesting one. You might think it would have an impact. In our epidemiological studies with colleagues in the UK and throughout the world, we've asked the question, does the routine vaccinations that infants and children already receive have an impact on risk? And with the possible exception of one vaccine, which is Haemophilus influenzae, there's no impact at all. So that's something different. That's not the normal microbiome. The way in which our immune network system, a complicated set of cells, responds to these beneficial bacteria is different from the way it responds to pathological microbes. So I think vaccines in the conventional sense would not work. I mean, you could argue if we're going to give infants a collection of natural bacteria, that is a kind of vaccine, but it's a very different vaccine from the ones that we give currently. So I wouldn't call it a vaccine.
So we are beginning to form a picture here. The gut microbiome most likely has a role in the development of all, but vaccinations don't. The next question then is, what are the implications of this hypothesis?
The bottom line here is that what this mechanism suggests to us is that the common form of acute lymphoblastic leukemia in children is probably preventable. And the line of thinking here is that we should be able to reconstitute what's called the normal microbiome, the normal gut microbiome of infants in a way that would be perfectly safe. So that's an exciting prospect. Before we can get to intervention in humans, we have to trial this in mouse models of leukemia, and that will be occupying my research team for the next 2 or 3 years. But the bottom line is that this common cancer is probably preventable.
Okay, so now we have an idea of the role the gut microbiome plays, but we wondered what other factors might be involved in the development of pediatric OL.
In cancer causation, we tend to look for, and perhaps anticipate, and I think the public anticipates, a singular explanation, like lung cancer is caused by cigarette smoke, skin cancer caused by ultraviolet light from the sun, and so on. But I don't think any cancer is as simple as that. They're all multifactorial, and almost all cancers are caused by a complicated mix of exposures, genetic inheritance, susceptibility, and background. And this is true of ALL. I mean, the chance aspect is complicated to explain, but there are biological explanations for that. But in childhood leukemia, as in, I think, all cancers, there is a genetic inherited component. And at the Institute of Cancer Research here in London, we've pinned down many of the genes involved in that. So that's no longer in doubt. The question is, or the uncertainty is, how much of the risk of leukemia in children is inherited and susceptibility. And I think that's a minor component, and I can't put a figure on it, but I think it's minor rather than major. And of course, there's nothing one can do about inheritance. So, but it is a factor, and even diet seems to have some indirect effect. So, as in all cancers, there are multiple components coming into the mix of risk. And what you look for is some major factor that really influences risk in a big way, and which is amenable to intervention. And the only thing here that's amenable to intervention is the infectious exposure component.
Currently, the cure rates for all are high, at around 90%, but the treatment is pretty toxic. So the idea of being able to prevent this disease is hugely exciting, and we asked Professor Greaves how he plans to take this research forward and what the next steps are.
Well, the focus is enormously now for me on demonstrating that childhood acute lymphoblastic leukemia is indeed preventable. I mean, that's the proof of the pudding, so to speak. I wish we could go straight into clinical studies, but that would not be practical because we have to work out exactly what mix of bacteria and what kind of preparation could be given, some kind of oral probiotic perhaps. What we have to do is to test this in mice. We have quite good mouse models now of acute lymphoblastic leukemia, whereby controlling the patterns of infection in the housing facilities, we can trigger acute lymphoblastic leukemia. So we have model systems in which we can show that we reconstitute, or we will be able to show, I hope, if we reconstitute the normal microbiome, it will be protective. If that works in mice, I think we then have a very strong case for pursuing this in clinical studies.
And in the meantime, is there any advice for parents at all? Any steps that they can take to perhaps reduce the risk for their children?
It's a very interesting question about what should parents do, and I also have been asked a lot about this recently. And it's a sort of difficult balance, because if you think about what's happened in modern societies of the past 100 years, we've cleaned up, we've sanitized our environments, we have better toilet facilities and engineering and cleaner water and so on. And that's all been incredibly beneficial because, in less developed societies and here 100 years ago, up to 20% of children would have died in infancy due to infectious disease. So it's fantastic we've done that. But the unexpected trade-off or consequence is we've deprived our infants of these beneficial bacteria. So the question is, how should parents treat their children to get the benefit of benign infections without incurring the penalty of the dangerous ones? Now, hygiene is still important, and washing hands is important, but we've probably taken it too far. But there are other aspects of society that aren't exactly hygiene that have reduced our exposure to beneficial microbes. I mean, the number of children in the house is important. The route of childbirth is important. Protracted breastfeeding is important. Insofar, before we get to an intervention or a vaccine type prophylactic, my advice would be not to be too super fussy about hygiene and take it too far. Allow children, don't get concerned if children pick up common bugs from other children. Just make sure they mix with other children. And breastfeeding for this and other reasons is obviously very beneficial.
The hygiene hypothesis that Professor Greaves alludes to may already be familiar to clinicians who follow research on type 1 diabetes, autoimmune diseases and allergy in young people. And on that note, we'll sign off. with a reminder that the article is published in Nature Reviews Cancer if you're looking for more details. A big thank you to Professor Greaves for speaking with us and to our listeners for tuning in. Until next time.